Welcome to the Deep Dive. Today, we're tackling, well, arguably one of the most intricate systems in the body, the endocrine system. Our mission is to really get to a mastery level using histology, the microscopic structure, as our guide. We'll go from the brain's command center right down to the glands regulating, everything.
Yeah, and using histology is absolutely the way to go. If you understand the structure, how the cells are put together, the function just clicks into place. The absolute core idea, the thing that defines this whole system, endocrine glands are ductless. Simple as that.
Ductless, meaning?
Meaning they're secretory cells, and these are almost always epithelial cells, interestingly enough. They release their signals, the hormones, straight into the surrounding tissue, right into the blood vessels. They're designed to dump product directly into nearby capillaries. Fast distribution.
And that distribution via the bloodstream is the classic definition, right?
Oh, yeah.
Acting on distant targets.
Exactly. That's the hallmark. These hormones circulate, and they act on target cells, maybe far away, cells that have very specific receptors just for that hormone. It's like broadcasting a signal, but only radios tuned to the right frequency can pick it up.
Okay, but here's a curveball you mentioned earlier. It's not just the big-name glands like the thyroid. This system is everywhere.
It really is. And that's often a surprise. You have critical endocrine cells tucked away inside organs that do completely different jobs. Think about the heart, pumping blood, or the kidneys, filtering waste, even the gut, digesting food. They all have specialized cells churning out hormones. It's a truly diffuse system.
All right, so this chemical communication, it's more complex than just long-distance calls with the blood.
Yeah.
You mentioned local signaling, too. We need to master those.
Absolutely essential, because a lot of regulation happens right there, locally. The first type is paracrine secretion. Think of this as like a neighborhood conversation. The signaling molecule just diffuses locally in the fluid between cells, or maybe through tiny short blood vessel loops right in that tissue.
Okay, a neighborhood chat. Give us an example.
The stomach is perfect. G cells in one part, the pylorus, make gastrin. But gastrin doesn't need to travel all over the body. It just diffuses locally to nearby cells in the fundic glands and tells them to secrete acid. It's targeted, efficient, right where it needs to be.
Makes sense. Then the next one requires actual physical contact, like a handshake.
That's a great analogy. Justicrine secretion. Here, the signal molecule doesn't even really get released. It stays stuck on the surface of the secreting cell, or maybe in the matrix right next to it. It only works when another cell physically bumps into it, makes direct contact.
Why have a system like that? Seems limiting.
Yeah, but that limitation is its strength. It's incredibly important during development when cells are migrating and deciding what to become, or in tissue repair. It ensures only the cell making direct contact gets the message. Very precise control over who listens.
And the third local type, the cell talking to itself.
Right, autocrine secretion. The cell makes a molecule that then binds to receptors on its own surface, or sometimes on identical cells right next door. Insulin-like growth factor, IGF, is a classic example. Cells make it, and that same IGF can then stimulate those very cells to grow or function. Self-regulation.
So we have different distances, different modes. What about the chemicals themselves, the hormones? They must vary a lot too. How do they interact with the target cell?
They fall into two broad chemical families, and it really dictates how they work. First, the hydrophilic ones. These are things like proteins, peptides, glycoproteins, basically water-loving molecules. They dissolve easily in blood.
But they can't cross cell membranes.
Exactly. They're water-soluble, not fat-soluble. So their receptors have to be on the outer surface of the target cell. The hormone binds, and that triggers a cascade of events inside the cell. It's like ringing the doorbell.
Okay, and the other type, the ones that can get inside?
Those are the hydrophobic hormones. Think steroids, like cortisol and thyroid hormones. They hate water, so they need special carrier proteins to travel in the blood. But because they're lipid-soluble, they can just diffuse right through the target cell's membrane.
So they don't need a doorbell?
Nope. They walk right in and bind to receptors inside the cytoplasm, or even in the nucleus. And they often work by directly affecting which genes get turned on or off. A totally different mechanism.
This is already incredibly complex. All these signals, near and far, different chemistries. How is it all controlled? How does the body keep it all in balance?
Through intricate feedback loops. This is absolutely key. Often, one endocrine gland releases a hormone that targets another endocrine gland. The second gland releases its hormone, which then circulates. When the level of that second hormone gets high enough, it signals back to the first gland, and maybe even higher up to the brain, telling it to stop releasing the initial stimulating hormone.
So it's self-regulating.
Precisely. It keeps hormone levels within very tight limits, prevents things from getting out of control, essential for homeostasis.
Okay, let's dive into the hierarchy then. Starting at the top, with the so-called master gland, the pituitary hypophysis. Tiny but mighty, right?
Tiny is right, about half a gram, tucked away in that little bony saddle in the sphenoid bone, the sella turcica. But its power comes from its amazing dual embryonic origin. You have to grasp this to understand the pituitary. It's essentially two glands fused together with different origins, different structures, different functions.
Glands in one. Let's break that down. Neural and oral.
Exactly. The neurohypophysis, that's the posterior part, is neural tissue. It actually grows down from the floor of the brain during development, from the diencephalon. It forms the main part, the pars nervosa, and the stalk connecting it to the brain, the infundibulum. Histologically, it looks like brain tissue because, well, it basically is an extension of the brain.
And the other part, the anterior pituitary, that's not from the brain.
Not at all. The adenohypophysis comes from the roof of the primitive mouth. An outpocketing of ectoderm called Rathke's pouch grows upward towards the developing brain. Its front wall thickens dramatically to become the main lobe, the pars distalis. There are also smaller parts, the pars tuberalis wrapping the stalk, and a thin pars intermedia between the anterior and posterior lobes. This fundamental difference in origin dictates everything about how they work.
So the connection to the hypothalamus, the brain region controlling it, must also be different for these two parts. How does the hypothalamus talk to the posterior pituitary?
It's a direct neural connection. It's called the hypothalamic hypophyseal tract. It's literally a bundle of axons, nerve fibers, running directly from nerve cells in the hypothalamus down the stalk and into the neurohypophysis.
Which nerve cells?
Primarily two clusters, or nuclei, in the hypothalamus. Neurons in the supraoptic nuclei, or S-zone, make antidiuretic hormone, ADH also called vasopressin. Neurons in the paraventricular nuclei, PVN, make oxytocin.
Wait, so the posterior pituitary doesn't make hormones?
Correct. This is a critical point for mastery. The posterior pituitary doesn't synthesize ADH or oxytocin. The hypothalamic neurons make them. The hormones are then packaged up, travel down the axons, and are stored in little swellings along the axons in the posterior pituitary.
What are those swellings called?
Neurosecretory bodies, or often herring bodies. You can see them under the microscope. They're like little storage depots. When a nerve impulse comes down from the hypothalamus, it triggers the release of the stored hormone directly from these herring bodies into nearby capillaries. It's neurosecretion.
Okay, direct nerve control to the posterior. But the anterior pituitary, the adenohypophysis, isn't directly connected by nerves, right? It came from the mouth roof. How does the hypothalamus control it?
This is where the system gets really elegant. It uses a specialized blood vessel network, the hypothalamic hypophyseal portal system. It's a brilliant piece of plumbing.
A portal system, meaning blood flows through two capillary beds in sequence.
Exactly. Arteries, the superior hypophyseal arteries, feed into the base of the stalk, the median eminence. There, they form a primary capillary plexus. These capillaries are fenestrated, they have little pores, they're leaky, and critically, this is where regulatory neurons from the hypothalamus terminate and release their hormone.
Tiny amounts of regulatory hormone.
Tiny amounts of neuropeptides, yes. Releasing hormones and inhibiting hormones. These leak into that primary plexus. Now, instead of going back to the general circulation, these capillaries immediately merge to form small veins, the portal veins, that travel a very short distance down the stalk. Then they break up again into a secondary capillary plexus that spreads throughout the anterior pituitary, the pars distalis. This portal system acts like a private hotline. It delivers those hypothalamic regulatory hormones directly and quickly to the anterior pituitary cells at a high concentration before they get diluted in the main bloodstream.
Ingenious. It ensures the anterior pituitary gets clear, strong signals directly from the brain.
Precisely. It's a key control mechanism.
Okay, let's zoom in on that anterior pituitary, the pars distalis. Histologically, how do we first classify the cells we see there? Based on staining.
Yeah, the traditional way is using stains. You see two main types of secretory cells called chromophils because they love stains. They pick up dye because they're full of hormone granules. They subdivide into acidophils, which grab acidic dyes like eosin, staining pink or red, and basophils, which prefer basic dyes, staining blue or purple.
And what about cells that don't stain well?
Those are the chromophobes. They look pale because they have few granules, maybe they've just released their hormones, or perhaps they're stem cells or precursor cells not yet actively secreting. It's kind of a mixed bag.
But the functional classification, based on what hormone they make, that's where the real understanding lies. Let's start with the acidophils.
Right. There are two main types of acidophils. First, the somatotrophs. These are the most common cell type in the anterior pituitary, maybe 50% of the total. They make somatotropin, which we usually call growth hormone, GH. GH drives growth mostly by stimulating the liver to produce other factors called insulin-like growth factors, or IGFs.
Okay, somatotrophs for growth. The other, acidophil.
Those are the lactotrophs, sometimes called mammotrophs, maybe 15–20% of cells. They secrete prolactin, PRL. Its main job is promoting milk secretion by the mammary glands after childbirth.
Got it. Now for the basophils, these seem to control many other endocrine glands.
They do. They produce the tropic hormones. First, you have the thyrotrophs, only about 5%. They secrete thyroid-stimulating hormone, TSH, or thyrotropin, which does exactly what its name says, stimulates the thyroid gland.
Straightforward. Next.
Next are the gonadotrophs, about 10%. Now here's a key detail for mastery. These cells are unusual because they secrete two different hormones: follicle-stimulating hormone, FSH, and luteinizing hormone, LH. Both are crucial for reproduction regulating follicle development, estrogen secretion, spermatogenesis, testosterone production, so on. But they come from the same cell type.
Two hormones from one cell type, interesting exception. And the last basophil type, the stress pathway starter.
The corticotrophs. About 15–20%. These guys make a very large precursor protein called pro-opiomelanocortin, or POMC. This big molecule isn't active itself. It gets chopped up inside the cell.
Chopped up into what?
Into several active peptides. The most important one is adrenocorticotropic hormone, ACTH, which stimulates the adrenal cortex. Another significant piece is beta-lipotropin, LPH, which is involved in lipid metabolism and can be further cleaved.
Why make one giant precursor like POMC just to cut it up immediately? It seems inefficient.
But it's actually very efficient for coordination. Think about a stress response. The body needs ACTH to stimulate cortisol, but it might also need other related signals simultaneously. By packaging them all into one precursor, the cell ensures that when the signal comes to release, all the necessary components are produced and secreted together in the right amounts at the same time. It guarantees a coordinated, multi-pronged response from a single gene product.
Clever. Okay, so these acidophils and basophils are the hormone factories, but they only work when told to by the hypothalamus via that portal system we discussed. What are those hypothalamic signals?
They are small peptides. They're releasing and inhibiting hormones. You have releasing hormones like TRH, thyrotropin-releasing hormone, which stimulates thyrotrophs, GnRH, gonadotropin-releasing hormone for gonadotrophs, GHRH, growth hormone-releasing hormone for somatotrophs, and CRH, corticotropin-releasing hormone for corticotrophs. These basically say go.
But there must be stop signals, too.
Absolutely crucial. The inhibiting hormones. The two main ones are somatostatin, which inhibits the release of both growth hormone and TSH, and dopamine, which acts as the primary brake, the tonic inhibitor, for prolactin release. The balance between releasing and inhibiting factors gives the brain fine-tuned control.
So that's the immediate top-down control from the hypothalamus.
Yeah.
What about the longer-term regulation? You mentioned feedback loops earlier. Can we walk through the thyroid example again?
Yeah. The negative feedback is key for stability. Let's trace it. 1, hypothalamus detects low thyroid hormone, TH, or needs more metabolic activity. It releases TRH. 2, TRH travels via the portal system to the anterior pituitary. 3, TRH stimulates thyrotrophs to release TSH. 4, TSH travels through the general circulation to the thyroid gland. 5, TSH stimulates the thyroid to produce and release thyroid hormone, T3 and T4. 6, TH circulates and acts on target tissues body-wide, but Q, crucially. 7, high levels of TH circulate back to the pituitary and hypothalamus and inhibit the release of TSH and TRH, respectively.
So the product shuts down its own production pathway?
Exactly. It prevents overshoot and keeps levels stable. Most endocrine axes work on this principle.
And when this goes wrong, say with a tumor in the pituitary,
then you can get serious problems. Pituitary adenomas are relatively common benign tumors. Usually they arise from one cell type and overproduce just one hormone. For example, a somatotroph adenoma. If that happens in a child whose growth plates haven't closed yet, the excess GH causes gigantism.
And in an adult.
If it happens after the growth plates fuse, you get acromegaly. Bones can't get longer, but they get thicker, especially in the hands, feet, and face. Soft tissues also grow. It really shows the power packed into those pituitary cells.
Okay, let's shift gears to the back door. The neurohypophysis, the posterior pituitary. We know it stores and releases ADH and oxytocin made in the hypothalamus. What does it actually look like histologically?
Well, it's basically neural tissue, remember? So you see lots of unmyelinated axons coming down from the SO and PVN in the hypothalamus. You also see those herring bodies, the storage depots, which look like pink blobs, but the most numerous cell type actually isn't neurons.
What is it then?
Specialized glial cells called pituitary. They look a bit like astrocytes in the brain. They're highly branched and wrap around the axons and nerve terminals.
What do the pituitarides do? Just support.
Primarily support, yes.
Mhm.
They physically surround the axon terminals where hormones are stored in herring bodies. It's thought they might help regulate the release of hormones into the capillaries, kind of like gatekeepers modulating access to the bloodstream.
And release is triggered by.
A nerve impulse. Pure and simple. An action potential travels down the axon from the hypothalamus, hits the terminal in the posterior pituitary, and causes exocytosis, the release of the hormone, which is bound to its carrier protein, neurophysin, from the herring body into the fenestrated capillaries there.
Let's quickly recap the hormones, ADH or vasopressin. What triggers its release and what does it do?
ADH release is triggered mainly by increased blood concentration, basically dehydration, detected by osmoreceptors in the hypothalamus. Its job is to go to the kidneys, specifically the collecting ducts, and make them more permeable to water. This allows the kidneys to reabsorb more water back into the body, concentrating the urine and conserving water.
Crucial for water balance.
And
Oxytocin.
oxytocin has two classic roles. It powerfully stimulates the smooth muscle of the uterus to contract during childbirth. It also triggers the contraction of myoepithelial cells around the milk ducts in the mammary gland, causing milk ejection, the letdown reflex during nursing.
But you also hear about other effects. Social bonding.
Yes, that's a really interesting area of research. Oxytocin also acts within the brain itself, and it seems to be involved in complex social behaviors, promoting feelings of trust, bonding between partners, and maternal behaviors. It's much more than just uterine contractions in milk.
And if the posterior pituitary system fails, like if ADH isn't released properly?
That leads to diabetes insipidus. Nothing to do with sugar diabetes or diabetes mellitus. Insipidus means tasteless, referring to the dilute urine. Without ADH, the kidneys can't reabsorb water effectively. So the person produces huge volumes of very dilute urine that's polyuria. And consequently, they experience intense thirst polydipsia. It can be caused by damage to the hypothalamus or pituitary stalk, tumors, or sometimes genetic defects.
Okay, that covers the central command. Let's move out to the periphery, the adrenal glands sitting on top of the kidneys. Another dual organ, right?
Absolutely. Two distinct parts fuse together. The outer layer, the adrenal cortex, develops from mesoderm and makes steroid hormones. The inner core, the adrenal medulla, comes from neural crest cells, the same origin as sympathetic neurons, and makes catecholamines, like adrenaline.
And you mentioned their blood supply is really special.
It's critical to how they function together. Arteries supply the outer capsule, then branch. Some arterioles punch straight through the cortex to supply the medulla directly with fresh arterial blood. But most arterioles form a network of leaky capillaries, sinusoids, throughout the cortex first.
So blood flows through the cortex before reaching the medulla.
A significant portion does, yes. This cortical blood, now rich in steroid hormones from the cortex, drains into the medullary capillaries and sinuses. So the medulla gets a dual blood supply, some direct arterial blood, and some venous blood that's already passed through the cortex and picked up cortical hormones.
Why is that important? What's the point of bathing the medulla in cortical blood?
It's about teamwork. The final step in making epinephrine, adrenaline, the main hormone of the medulla, requires an enzyme, PNMT, whose synthesis is actually stimulated by glucocorticoids, like cortisol, coming from the cortex. So by receiving cortisol-rich blood from the cortex, the medullary cells are primed and ready to produce maximal amounts of epinephrine when the stress signal hits. It's a clever anatomical link ensuring functional synergy.
Very neat. Okay, let's look at the cortex histology. These cells make steroids, which are lipids. What features do we look for?
Steroid-secreting cells have a characteristic look. They often appear pale or foamy because they contain lots of lipid droplets, which get washed out during tissue prep. Their cytoplasm stains pink, acidophilic, but the key organelles are inside. They have an incredibly extensive, smooth endoplasmic reticulum, SCR, because that's where many steroid synthesis enzymes are. And their mitochondria are unusual. Instead of flat shelves, their inner membranes form tubes, tubular cristae, which are packed with other enzymes needed for steroid production.
And they don't store the hormones.
No, steroids are small and lipid soluble. Once made, they just diffuse right out of the cell into the blood. No granules needed.
The cortex has layers or zones, 3 of them, from outside in.
Correct. Just under the capsule is the zona glomerulosa, makes up about 15%. The cells here are arranged in little rounded clusters or arches, kind of like glomeruli. Their job is to make mineralocorticoids, primarily aldosterone.
Aldosterone, that's for salt balance.
Exactly. It acts on the kidney tubules to make them reabsorb sodium and water follows and secrete potassium. It's crucial for maintaining blood pressure and electrolyte balance. And importantly, its main controller isn't the pituitary. It's mostly regulated by the renin-angiotensin system in the kidney and by blood potassium levels. ATTH from the pituitary has only a weak effect here.
Okay, moving inward to the thickest layer.
That's the zona fasciculata, maybe 65–80% of the cortex. Fasciculata means bundles. The cells here are arranged in long, straight cords, like columns, separated by capillaries. These cells are often quite large and look very foamy or spongy because they are packed with lipid droplets.
MMA.
Glucocorticoids, with cortisol being the main one in humans. Cortisol has widespread effects. It raises blood glucose, suppresses the immune system, helps manage stress, mobilizes fats and proteins, and this zone is tightly controlled by ACTH from the pituitary. It's the core of the HPA axis, hypothalamic pituitary adrenal axis.
And the innermost cortical layer, right next to the medulla.
The zona reticularis, about 10%. The cells here form irregular, interconnected cords. Reticular means network-like. They tend to be smaller, stain a bit darker, have fewer lipid droplets, and often contain a brownish wear-and-tear pigment called a lipofuscin.
Their main product.
They produce some cortisol, but primarily they secrete weak androgens, like DHEA, dihydroepiandrosterone. These aren't very potent themselves, but can be converted to testosterone or estrogen in other tissues. Like the fasciculata, the reticularis is also stimulated by ACTH.
And if the entire cortex fails, like in Addison disease.
That's serious. Addison disease, often autoimmune, destroys the cortex. Loss of aldosterone from the glomerulosa causes dangerous salt wasting, low blood pressure, and high potassium. Loss of cortisol from the fasciculata causes metabolic problems, weakness, and inability to cope with stress. It highlights how vital these zones are.
Now let's plunge into the core, the adrenal medulla. Neural crest origin, making catecholamines. What are the cells called?
They're called chromaffin cells. Large, kind of pale staining cells, often arranged in clusters. Remember, they're essentially modified sympathetic postganglionic neurons. They just lost their axons and became specialized secretory cells.
And they store their hormones, unlike the cortex.
Yes, they store catecholamines, mainly epinephrine, about 80%, also called adrenaline, and norepinephrine, about 20% noradrenaline, in dense cord granules bound to proteins called chromogranins. You can see these granules with electron microscopy.
How is their release controlled? Not by ACTH.
No, this is direct neural control. Preganglionic sympathetic nerve fibers come directly from the spinal cord and synapse right onto the chromaffin cells. When the sympathetic nervous system fires up fight or flight, these nerves release acetylcholine, which triggers the chromaffin cells to dump their stored catecholamines straight into the bloodstream. It's incredibly fast.
And the effects, fight or flight responses.
Pretty much. Epinephrine increases heart rate, blood pressure, mobilizes glucose, dilates airways, shifts blood flow to muscles. Norepinephrine strongly constricts blood vessels in many areas. Together, they prepare the body for immediate, intense physical action.
In tumors here, pheochromocytomas.
Right. These are tumors of the chromaffin cells. They can periodically release massive amounts of catecholamines, causing wild swings in blood pressure, headaches, sweating, palpitations. It really demonstrates the power of the medulla's hormones.
Okay, moving on. Another gland that mixes functions. The pancreas. Mostly exocrine, but with crucial endocrine islands.
Exactly. Scattered throughout the exocrine tissue that makes digestive enzymes are the islets of Langerhans. These are little compact clusters of endocrine cells, maybe making up only 1–2% of the pancreas volume, but absolutely vital for regulating blood sugar.
What do they look like structurally?
They're like little balls of cells arranged in cords and riddled with fenestrated capillaries for quick hormone release into the blood. What's really key for mastery here is understanding that the different cell types within the islet aren't randomly mixed. Their position matters.
Position matters. Why?
Because they talk to each other locally using paracrine signaling in addition to releasing hormones into the blood. The main cell types are beta, alpha, and delta cells.
Okay, tell us about them and where they are. Beta cells first.
Beta cells are the most numerous, maybe 70%. They are typically located in the core, the center of the islet. They produce insulin. Insulin's main job is to lower blood glucose by helping cells take it up and store it.
And alpha cells?
Alpha cells make up about 20% and are usually found around the periphery of the islet. They secrete glucagon. Glucagon does the opposite of insulin. It raises blood glucose, mainly by telling the liver to break down stored glycogen and release glucose.
So beta in the middle lowering sugar, alpha on the outside raising it. What about the delta cells?
Delta cells are less common, 5–10%, and they're kind of scattered within the islet. They secrete somatostatin. Now, remember somatostatin from the hypothalamus inhibiting GH and TSH? Here in the islet, it acts locally to inhibit the release of both insulin from the beta cells and glucagon from the alpha cells. It's like an internal brake within the islet, helping to fine-tune the output.
A local regulator.
Yeah.
Clever. Were there other cell types mentioned? PP cells?
Yeah, PP cells, or F cells, are rare in most islets, but more common in islets located in the head of the pancreas. They make pancreatic polypeptide, which seems to influence gut motility and some digestive secretions. Less central to glucose control, though.
So the islet is regulated primarily by blood glucose itself, but also by this internal paracrine chatter and external nerves, too.
Absolutely. High blood glucose is the main trigger for insulin release from beta cells. Low glucose triggers glucagon from alpha cells. But the autonomic nervous system is wired in too. Sympathetic stimulation, stress, fight or flight, tends to increase glucagon and decrease insulin. You want sugar available. Parasympathetic stimulation, rest and digest, tends to increase both, helping store nutrients after a meal. It's a really complex interplay.
And when this intricate system breaks down, we get diabetes mellitus.
Correct. In type 1 diabetes, it's usually an autoimmune attack that destroys the beta cells in the core of the islets. No insulin can be produced. In type 2 diabetes, which is much more common, the beta cells are still there, but either the body's tissues become resistant to insulin's effects or the beta cells just can't produce enough insulin to cope with the demand, especially in the context of obesity. Either way, glucose control is lost.
Let's move up to the neck, the thyroid gland. You said this one has a unique storage system.
Utterly unique in the endocrine world. The thyroid is made up of millions of tiny spherical structures called thyroid follicles. Each follicle is a ball of simple cuboidal epithelial cells, the thyrocytes, surrounding a central space, a lumen, that's filled with a pink gelatinous goo called colloid.
And that colloid is the stored hormone.
Not quite the hormone itself, but the precursor. The colloid is mostly thyroglobulin, a huge protein made by the thyrocytes. The thyrocytes attach iodine atoms to this thyroglobulin and store massive amounts of it in the colloid extracellularly. This is the unique part. Storing the precursor outside the cells allows the thyroid to keep weeks, even months, worth of hormone supply on hand. No other endocrine gland does that.
Incredible storage capacity. Are there other endocrine cells in the thyroid besides the follicle cells?
Yes, tucked in between the follicles, or sometimes within the follicle wall but not reaching the colloid, are the parafollicular cells, or C cells. These have a different origin neural crest, not the pharynx like thyrocytes. They look larger, paler staining. They make a completely different hormone, calcitonin.
Calcitonin.
Yeah.
That lowers calcium.
It does, primarily by inhibiting osteoclasts, the cells that break down bone. However, in humans, its role seems relatively minor compared to the main calcium regulators. It's not considered essential.
Okay, back to the main event. Making thyroid hormone, T3 and T4. This sounds complex, involving both making stuff to put into the colloid and then taking it back out, all driven by TSH.
Exactly. It's a fascinating multi-step process, like a cellular assembly line switching between export and import. 1. Make the scaffold. Thyrocytes synthesize that huge thyroglobulin protein in their RER and Golgi, then secrete it out into the colloid. That's like building the framework. 2. Import the raw material. They actively pump iodide ions from the blood using a special transporter called NIS, sodium iodide symporter. They concentrate iodide like crazy, maybe 30 times the blood level, then they transport it into the colloid. 3. Activate and attach. At the cell surface facing the colloid, an enzyme called thyroid peroxidase oxidizes iodide, IOR, to reactive iodine and immediately attaches it to tyrosine amino acids that are part of the thyroglobulin molecule already floating in the colloid. 4. Couple them up. Still within the colloid, the enzyme links pairs of these iodinated tyrosines together to form the actual hormones, T3, triiodothyronine, 3 iodines, and T4, thyroxine, 4 iodines, still attached to the thyroglobulin backbone. 5. Bring it back in. When TSH from the pituitary signals, the thyrocytes reach out and engulf droplets of this iodinated thyroglobulin from the colloid via endocytosis. 6. Liberate and release. These vesicles fuse with lysosomes inside the cell. Lysosomal enzymes chop up the thyroglobulin, releasing the finished T3 and T4 hormones. These small hormones then diffuse out the back of the cell at basal side into the capillaries.
Wow. Exocrine secretion into the colloid, then endocrine retrieval and release into the blood. Amazing process. And TSH drives pretty much all of it.
TSH stimulates nearly every step: iodide uptake, thyroglobulin synthesis, iodination, endocytosis, proteolysis. It's the master switch.
Which explains why problems arise with iodine deficiency or TSH issues.
Precisely. If you lack iodine, you can't make T3, T4 properly. Steps 3 and 4 fail. Low T3, T4 means less negative feedback, so the pituitary pumps out more TSH. This excess TSH constantly stimulates the thyroid cells, steps 1, 2, 5, causing them to grow and fill the follicles with useless uniodinated thyroglobulin. The gland enlarges massively, that's a goiter. Or in Graves' disease, you have autoantibodies that mimic TSH, constantly stimulating the gland, leading to hyperthyroidism, overproduction of T3, T4.
Okay, nearby, usually stuck to the back of the thyroid, are the tiny parathyroid glands. What's their main job?
4 tiny glands, sometimes fewer, sometimes more, sometimes in weird locations. Their mission is singular and critical: regulate blood calcium levels, specifically to raise blood calcium when it gets too low.
What are the main cells doing the work?
The vast majority are principal cells, often called chief cells. They're small, polygonal, relatively pale staining. These are the cells that sense low blood calcium and secrete parathyroid hormone, PTH, in response.
Are there other cell types?
Yes, you also see spattered clusters of much larger, very pink-staining acidophilic cells called oxyphil cells. They're packed with mitochondria, but their function is unclear. They might be inactive or degenerating chief cells. They definitely increase in number as we age. But PTH comes from the chief cells.
And PTH raises calcium. How? Where does it act?
PTH has 3 main targets. 1, bone. It indirectly stimulates osteoclasts, the bone-dissolving cells, via signals from osteoblasts. More bone breakdown releases calcium and phosphate into the blood. 2, kidney. It directly tells the kidney tubules to reabsorb more calcium from the filtrate so less is lost in urine. It also causes the kidney to excrete more phosphate, which helps prevent the released calcium and phosphate from just precipitating out again. 3, intestine. This is indirect. PTH stimulates the kidney to perform the final activation step for vitamin D. Active vitamin D is essential for absorbing dietary calcium from the gut.
So bone, kidney, gut, a 3-pronged approach to boost blood calcium. What happens if PTH levels are wrong?
If you have too little PTH, hypoparathyroidism, blood calcium drops dangerously low. This can cause muscle cramps, twitching, even seizures, tetany, because calcium is crucial for nerve and muscle function. Bones might become denser but more brittle. If you have too much PTH, hyperparathyroidism, blood calcium gets too high. The excess calcium is pulled from bones, making them weak, and it can deposit pathologically in soft tissues, like forming kidney stones or calcifying arteries. Not good either way.
All right, one more distinct gland to cover. The pineal gland, deep in the brain. What's its story?
Tiny pinecone-shaped structure, hence the name. Develops from the roof of the third ventricle, so it's neuroectoderm origin. Its main job is regulating circadian rhythms, our daily sleep-wake cycles.
And the main cell type?
Pinealocytes. These are the secretory cells that produce the hormone melatonin, which is actually derived from the amino acid tryptophan, similar to serotonin.
How does it regulate rhythms? It's often called a neuroendocrine transducer. What does that mean?
It means it converts a neural signal into a hormonal signal. Specifically, it converts information about light and darkness into melatonin production. Light hitting the retina sends signals via a complex pathway, retinohypothalamic tract to sympathetic nerves, that ultimately inhibits melatonin release. Darkness removes that inhibition, so melatonin levels rise at night. This nightly surge of melatonin helps regulate sleep patterns and other cyclical body functions.
So it translates darkness into a sleepy hormone signal. Any weird histological features here? Brain sand.
Ah, yes, corpora arenacea, or brain sand. These are little calcified concretions like tiny pebbles of calcium and magnesium salts that accumulate within the pineal gland as we age. They don't seem to do anything functionally, but because they're calcified, they show up clearly on X-rays and CT scans. Radiologists actually use them as a handy landmark to check if the midline structures of the brain have shifted due to a tumor or swelling.
Useful, even if non-functional. Okay, we've covered the major glands. But you mentioned endocrine cells are scattered everywhere, the diffuse system.
Right, the diffuse neuroendocrine system, or DNES. This isn't a single gland, but a vast population of individual endocrine cells scattered throughout the body, especially lining the gut, where they used to be called enterochromaffin cells, and the airways. You also find some in the pancreas outside the main islets.
What makes them neuroendocrine?
Because many of them produce the same signaling molecules, peptide hormones, or amine neurotransmitters like serotonin that are also found in the central nervous system. They share a common chemical language.
There were some old, confusing names for these cells.
Yeah, historically, names like argentaphin or gyrophil or APUD cells were used based on staining properties or biochemistry. Now, it's more common to classify them by the specific peptide they secrete, like G cells for gastrin, S cells for secretin, etc. The key takeaway, though, is their function.
Which is
Primarily paracrine, acting locally. Because they're scattered right there in the gut lining or airways, they can quickly release signals that regulate local blood flow, muscle contractions, motility, and secretions from nearby exocrine glands. Very important for fine-tuning digestion and respiration on the spot.
Okay, let's try to pull all these threads together. We've seen a central command, hypothalamus pituitary, glands for stress and salt, adrenals, metabolism, pancreas, thyroid, calcium, parathyroids, and rhythms, pineal.
Exactly. And the beauty is how interconnected they are. Think about ACTH from the pituitary controlling two different zones in the adrenal cortex making different steroids, or somatostatin from delta cells in the pancreas inhibiting both alpha and beta cells right next door. It's all about layers of control and feedback.
The structural variety was amazing too. The follicles of the thyroid, the cords and clusters in the adrenals and pituitary, the scattered islets in the pancreas. Yet all fundamentally endocrine ductless epithelial origin mostly, using the bloodstream.
And achieving real mastery means appreciating how the signals are delivered. That mix of long-distance endocrine signals, local paracrine whispers, cell-to-cell juxtacrine nudges, and direct neurosecretion. The system uses whichever method is best suited for the speed and target required.
Which brings us to a final thought for you, our listener, to ponder. We often think of the endocrine system as, well, relatively slow. Hormones circulate, feedback loops take hours or days, like with the thyroid or the HPA axis managing cortisol.
But think about the adrenal medulla. Direct nerve input causes an instant flood of adrenaline. Think about the pineal gland converting the immediate presence or absence of light into melatonin signals. How much of our moment-to-moment physiology, our sleepiness, our sudden alertness, is driven by these instantaneous neuroendocrine responses, bypassing those slower loops entirely? How is that lightning-fast neural-to-hormonal switch fundamentally different from the more gradual systemic feedback that governs so much of our metabolism? It's a fascinating contrast in timing and control within the same system.